In this video, we'll be discussing one of the aspects of genetics and biotechnology, and that's recombinant. Let me switch markers. Recombinant DNA, which is DNA made from two or more sources. So we've taken DNA maybe from two different animals or two different bacteria or two or more different organisms. We've taken their DNA and we've recombined them into a new piece of DNA. And essentially, building recombinant DNA is just cut and paste on a molecular level. And in order to cut that DNA, we use restriction enzymes. And they cut DNA at specific sites. And we call those sites restriction sites. And there are hundreds of known restriction enzymes that all cut DNA at different sequences. And so if we know the sequence of the DNA that we want to cut, we can, so if this is our DNA, then we can take that sequence, enter it into a computer program, and it will run along that sequence, and it will identify, this one can be cut by the enzyme EcoR1. This could be cut by EcoR5. This could be cut by BAMH1. This could be cut by HindD3. And these are all different restriction enzymes that I have used when working with DNA. And there are hundreds of others, and each one recognizes a specific sequence of DNA and cuts the DNA at that sequence. And these restriction enzymes, some of them will cut right down the middle. And so if this is our DNA, some restriction enzymes will cut right down the middle and leave two ends called blunt ends. And blunt end restriction enzymes are a little tricky to work with compared to the other type of restriction enzyme that instead of cutting right down the middle, kind of does this. And these are called sticky ends. Because the sticky ends, we have a short segment of single-stranded DNA that sticks out from each piece of DNA after the cut. And these pieces can base pair with each other. They can grab onto each other, and then we can reconnect those pieces. And so because they stick together, then we have more control over how the DNA, over how our recombinant DNA will be built. Because after we cut the DNA with the restriction enzymes, we use an enzyme called ligase to glue the DNA pieces together. And so this is essentially the cut and paste. The cut comes from restriction enzymes. The paste comes from the ligase. And I'll try to explain this with another diagram. So let's say there's a certain gene from an animal that we want to study. That gene makes a particular protein. We can get that protein in one of two ways. What they used to do was to take a large amount of tissue samples from an animal, which required possibly getting lots of individual animals, killing all of them, dissecting them, taking the specific tissue out of each, grinding it up, separating the proteins chemically, and after you purify the protein, you might have a very small amount of the stuff to study. Or you take the DNA from the animal, which you can take a DNA sample from a living animal pretty easily and without doing any real harm to the animal. You take that DNA, you take a specific gene for your specific protein, you put that gene into bacteria, the bacteria produce the protein instead of the animal, and now you can grow up a huge amount of bacteria and make a huge amount of protein, and you can study that protein without killing any animals. And so it's a lot more convenient than what they used to do when isolating proteins. And so if we have, let's say this is a piece of DNA from the animal whose protein we want to study. And it has two restriction sites. We'll say restriction site A and restriction site B. And so it'll cut the DNA in these two pieces, or at these sites that will digest the DNA with both A and B to cut out this piece. And this piece contains the gene for the protein we want to make. And so now what we need is a small piece of bacterial DNA. Remember, the bacterial DNA is usually circular, and the small circular DNAs from bacteria are called plasmids. Well, this is only one plasmid, so I'll erase that S. And this plasmid might also contain restriction sites for A and B. And so we'll cut the plasmid with the same A and B enzymes that we use for our animal DNA. And so that will leave that will open up the plasmid. We'll cut it open, and now we have two pieces of DNA. This piece that contains the gene we want to study, and this bacterial DNA left over from the plasmid. And so we'll combine, we'll mix these two together, and those sticky ends will cause it to associate with each other. And so the two pieces of DNA will bind together, and then we use a ligase to glue them to each other so they can't fall apart again. And so now we have this plasmid that contains this piece of animal DNA that codes for a certain animal protein. And at this point, if we want to make more of this DNA, it's real easy. We just put it into a bacteria. And because it's bacterial DNA along this plasmid backbone, the bacteria will accept that DNA and make more of it. And as the bacteria grow and divide, they make more and more of this DNA. And so you can grow up several liters of a bacteria and produce large amounts of this DNA that we can then purify from the bacteria and get large amounts, however much DNA we need to do our experiments. If we need to make protein, we can put this into a different type of bacteria that's been engineered to make large amounts of protein, and the bacterial enzymes will transcribe the gene to make mRNA. The mRNA will be translated to make protein, and the bacteria can produce lots and lots of protein that we can purify from the bacteria, and we can get however much protein we need to do our experiments. That this is used not just for experimental biology and biochemistry. It has, the recombinant DNA technology has practical use in society. And one of those is insulin. That before the advent of recombinant DNA technology, insulin for diabetics was produced from pig or cow pancreas. And so the slaughterhouses would take the pancreas out of the animal while they were cutting everything up, and you'd have to collect all of those pancreases and grind them up and process them to get the insulin. And the insulin was very expensive, and it wasn't human insulin. It was a little bit different from human insulin. And so it would occasionally cause side effects and immune system problems for the diabetics who needed it. But using recombinant DNA technology, we were able to take the DNA for human insulin, we were able to stick that into bacteria, and we can use the bacteria to produce tremendous amounts of human insulin that can be safely injected into a person, much cheaper and much cleaner than producing that insulin from extracting it from pig or cow pancreas. Another example would, if you're familiar with the farms around here, would be Roundup Ready corn plus soybeans. That the herbicide Roundup used to be used to kill just about every plant out there. And so it wasn't particularly useful because it killed everything. But using some genetics and some genetically modified organisms, they were able to engineer corn and soybeans that contain an enzyme that allows the plant to continue surviving in the presence of the Roundup molecule, which is the active ingredient in Roundup is called glyphosate. And glyphosate inhibits the production of aromatic amino acids like phenylalanine, tryptophan, and tyrosine, which are necessary for the plant to survive. And so the genetic engineering allows the plant to continue to produce those aromatic amino acids even when exposed to glyphosate. And so the corn and soybeans will survive because they're genetically engineered, but the weeds will not. And so now you can use you can spray your corn or your soybeans with Roundup, and you'll kill all the weeds, and your crops will survive. Another useful example for scientific research is GFP, or green fluorescent protein. This is a gene that was taken from jellyfish that when you hit GFP with ultraviolet light, then it will fluoresce. It will glow a bright green color. And so we can take this GFP and let's say we want to study the location of a particular protein inside a cell. Well, we can make what's called a fusion protein. And a fusion protein is when we take the genes for two proteins and we stick them together so that it makes a single messenger RNA and it makes a single protein, and that single protein is made up of two proteins that are stuck together. And so we take this GFP gene and we attach it to the gene for whatever protein we want to study inside a cell. And now that fusion protein is fluorescent. And we can use a fluorescent microscope to watch where these proteins go inside the cell. And so we can follow our proteins around the cell by looking for this GFP green fluorescent protein. A similar protein, or it's not a similar protein, but it's used for a similar function, is luciferase. And luciferase is an enzyme from fireflies. It's the enzyme that allows fireflies to light up. That it takes a chemical called luciferin, and it breaks that luciferin down, and it releases light during that chemical reaction. And so we can take the gene for luciferase out of fireflies, and we can put it into other animals. And that if this is a firefly, and it lights up, well, we can take its DNA and we can stick it into a mouse, and this is my highly accurate mouse, and we can make that mouse light up. And this was a lot of my research as a graduate student was taking this luciferase DNA and trying to insert that into mice and then measuring how much light the mouse tissues could produce using a technique called bioluminescent imaging. And so it doesn't make a lot of light. We cannot see it with the naked eye, but with a sensitive camera, we can see the mice light up. And normally, if we do the technique right, we anesthetize the mice so they don't move around when we're taking the picture, because it's a very long exposure time. You have to take the picture over about a minute. At one time, I did the experiment without anesthetizing the mice, and so they woke up and started walking around while we were taking the picture. And so instead of getting a mouse and measuring the light from the mouse, all we saw was this green path as the mouse was walking around while we were taking the picture. That was pretty funny. But the recombinant DNA technology has allowed us to do incredible amounts of scientific research into how proteins work, how genes are regulated. We can use it to study. We can use GFP and luciferase to study like cancer in mice. The applications for recombinant DNA technology are almost endless. And it's becoming easier all the time. That process of cutting and pasting DNA might be relatively obsolete, because now there are companies that you just send them a computer file with a sequence of DNA that you want to make, and they will chemically construct that DNA one base at a time, and they'll send you a piece of DNA that you can stick into a plasmid, or sometimes you can buy it directly inserted into a plasmid already, and so you don't have to do any of that cut and paste work. You can just buy the DNA ready to go. And so the recombinant DNA industry is changing very rapidly, and is very important for biology and biochemical research.